Dear student, today we will discuss about agarose gel electrophoresis. Now, what do you mean by electrophoresis? Electro means charge and phoresis means migration. Therefore, the migration of charged particle under the influence of electricity is termed as electrophoresis. Gel electrophoresis is the laboratory technique used to separate DNA, RNA, or protein molecule based on their size and electrical charge. Agarose gel electrophoresis is used for separation of DNA and RNA fragments, whereas polyacrylamide gel electrophoresis is used in case of proteins. So there are so many types of electrophoresis, and one of them is the gel electrophoresis. And again, you see in the gel electrophoresis we have agarose gel and polyacrylamide gel. So it depends what separation we are interested in. So for DNA we use the agarose gel electrophoresis. Let's see what is the principle behind this process. The DNA is negatively charged due to presence of phosphate backbone. So when it is allowed to run on the agarose gel electrophoresis, it will move toward the positive electrode. The net charge on DNA fragment and their size will decide the movement on gel. The porosity of gel allows small molecules to move faster and farther as compared to the larger one, which moves slowly and to the shorter distance. Different size fragment of DNA separation is achieved on the gel and visible by staining with suitable dye. So the DNA is visible in the form of band. Now we will see what are the requirement and how this is done. So the requirement is first the electrophoretic apparatus, which consists of gel cassettes, electrophoretic tank, comb and power supply. So cassette is used to cast the gel. An electrophoretic tank is to carry out the whole process of electrophoresis. Comb is used to make the wells, and power supply is to provide the charge. So next requirement is the agarose. Agarose is known as molecular sieve in molecular biology. It is actually polysaccharide which is obtained from seaweed, and this is available in the dry powder form from which the preparation is done. So from the market it is available in the dry powder form. So how we prepare the gel from the dry powder? So the powder is taken to this, the buffer, electrophoretic buffer is added. So a solution is made and now this is heated. So after heating, the molten solution of agarose gel is formed, and this is allowed to pour in the cassette, the gel cassette. Okay, and then before cooling you have to place the comb. So the comb is placed in the molten state of gel. Now this is allowed to cool down, solidify. So once it gets solidified, slowly you will remove the comb and the wells are created in the gel. So these wells are used to load the sample. Now we will see the buffer, the type of buffer that is used in the electrophoresis. So tris borate EDTA or TBE buffer is used. EDTA means ethylene diamine tetraacetic acid is a chemical, is known as chelating agent, and it binds to divalent cations such as magnesium, calcium, and prevent the degradation of DNA. So how it prevent the degradation of DNA? The EDTA, it bind to divalent cation. These divalent cation is actually required by the enzyme nuclease, which are DNAs, like DNAs or RNAs. So DNAs act on the DNA and it degrade the DNA. So the requirement is calcium and magnesium. So when these calcium and magnesium are not made available to DNAs, then they cannot do their function, and in this way they prevent the degradation of DNA. Secondly, the buffer maintains the neutral pH. Thus, it controls the net charge of molecule, and that helps in the proper migration and separation of molecule. Next is we use the diethidium bromide for staining of DNA. and this is added to either this is added to the DNA while loading the sample, or once the DNA fragments are separated, the later on staining is done, because the purpose is to visualize the DNA. This is intercalating dye and get inserted in the double stranded DNA between the bases. It is fluorescent dye that has UV absorbance of 300 to 360 nanometer, and the DNA is visible in the form of band. Okay, and the instrument which is used to make it visible is called trans illuminator or fluorimeter. Now, what are bands? Each band has number of fragments of DNA. So ultimately they represent the size of the DNA. And next is the sample DNA. Sample is the DNA to be examined. The DNA after extraction from the source is stored in the buffer, and now this is ready to be used as sample DNA. Then we also use the marker DNA, which is a standard, and it consists of fragment of different DNA size, known DNA size for the comparison. So let's see the methodology. So here we have the electrophoretic tank. So you have to place your gel in the electrophoretic tank and flood this tank with the buffer. Now you have to load these samples. So in different wells, different samples are added, and then marker DNA marker is also added. So in the first well we add the DNA marker, whereas in the others we um, at the different samples to be examined. So the loading of sample is done with the help of micropipette. Now you have to connect. Now once the sampling is done, we have to connect it with the power supply. The DNA will run from negative to positive terminal, and once the DNA migration has taken place, the gel is taken out for staining. Now what we do is do the staining. So Ethereum bromide solution is prepared, and in that the gel is kept for certain period of time. Now the gel is ready to be washed under the UV illuminance. So you will see the bands in the marker. You will see the different types of different fragments of DNA of different sizes are there, and then different sample have different size DNA. You can always compare it with the marker and find out that your sample number 1 has the fragment of 2500 kb, and so on. Okay, so this DNA analysis is done in case of DNA fingerprinting, in case of research purpose, and to diagnose the disease. So this is all about gel electrophoresis. Thank you.